Today, I'm very excited to share with you a project on the topic of personalized microbiome intervention. So we're all colonized by trillions of microorganisms called the microbiome, and they play a critical role in our health. So through different stages of life, infancy actually represents the most critical time for the formation of our microbiome. Disruption of the early life microbiome caused by events like premature birth or antibiotic administration could put children at a higher risk of developing chronic issues like diabetes and allergies. So a lot of audience here already know that breast milk is considered to be the most ideal food sources for babies because it is enriched with a lot of functional compounds like human milk oligosaccharides, which are a group of fibers that are unique to breast milk, and they selectively enrich certain beneficial gut microbes like Bifidobacterium and can ensure a proper development of the infant gut microbiome. These days, one or more HMOs are often added to infant formula in order to recapitulate some beneficial effects of breastfeeding. However, it has been reported in the past that different infant microbiomes would respond to the same HMO differently. And to this day, we still lack a clear understanding on why such variation happened. So here we picked a prevalent HMO, 2-fucosyllactose, shown here, and we investigated why different infant microbiomes may have different 2-FL metabolism. And to gain a mechanistic view of the microbiome 2-FL metabolism, we established in vitro infant gut microbiomes using poops directly sourced from babies. Specifically, we selected 3 early life stool samples from 3 different babies. 2 of them are full-term babies, FT1, FT2, and then the third infant is a preterm infant termed PT1. And as you can see, all of these samples are collected before 3 months of age, which is considered as the critical window. And the first thing we want to do is to figure out a growth condition that allow us to reproducibly and stably recover most species from the stool inocula. And then we landed on 3 base media, BHI, MGAN, and pooled breast milk. We also added mucin because it is a common carbon source in the gut. So we tested all these growth media using one sample, the FT1 sample, and I have shown you all the growth outcomes. This is an average from all the replicates. And long story short, what we found was that BHI with mucin supplementation with just one passage allowed us to recover all 9 species, and this growth condition is considered to be highly reproducible because all the replicates are actually quite similar to each other, and this growth condition is also considered to be stable. So to verify this growth condition, we tested 2 other infant samples, and the conclusions are similar. So once we figured out that BHI with mucin with one passage is the condition that we want, we next move to test how 2-FL affect these different infant microbiomes. So we tested 2 different concentrations. One is around the physiological level of 2-FL, which is 0.3% weight over volume. And then we also tested a concentration of 2-FL that's 10x lower. So what I'm showing you here is adding a tiny amount of 2-FL, and I'm showing you the composition of FT1, FT2, and PT1. And as you can see, all of them kind of look like the ones that were grown in just BHI and mucin. What is interesting is that when you add a 2-FL at 0.3%, you see a drastic differences. And specifically, I want you to pay attention to Bifidobacterium breve, which is this navy color here. And as you can see, in FT1, you have a drastic increase of B. breve abundance. For FT2, you see a moderate increase. And for PT1, you barely see any increase. And you can also see the differences at the pH level. So we have 2 hypotheses. One is that maybe there's genomic differences among the Bifidobacterium strains from these 3 infant samples. And the other hypothesis we have is that maybe there are different microbial interactions that lead to this different 2FL metabolism. So before I dive in, I just want to quickly show you that to metabolize 2FL, you need to have fucosidases, which break down 2FL into L-fucose and D-lactose. So the first thing we did was to just compare the Bifidobacterium strains from these 3 infant microbiomes. And what we found was that the 3 strains are actually highly similar to each other. And then we also look at the relevant gene content. And what we found was that the Bifidobacterium strains all encoded 1 copy of intracellular GH95 fucosidase, and none of these Bifidobacterium strains have any confident hits from the metagenomic search of 2FL transporters. So this led us to conclude that none of the Bifidobacterium strains can actually metabolize 2FL on its own. So this led to the second hypothesis. So what about the other community members? So now coming back to this figure that I showed you earlier, I know there are a lot of species here. I want you to focus on the ones that I highlighted. So besides the Bifidobacterium, the Ruminococcus gnavus, and also the 3 firmicutes from FT2 also encode fucosidases. But what makes these species special is that they also encode extracellular fucosidases, meaning that they can chop 2FL extracellularly and releasing the sugar to the growth environment. So this led us to have a new hypothesis, basically is that there's a species breaking down 2FL, and the sugar could be then picked up by Bifidobacterium for growth. So to test this hypothesis, we went back to FT1 and we isolated Bifidobacterium and the Ruminococcus gnavus, and we did a co-culture experiment. So here I'm showing you 3 separate panels of the growth in 2FL, L-fucose, or D-lactose as the sole carbon source. So when just growing as it by itself, the Bifidobacterium did not show any growth on 2FL or L-fucose. However, it grew extensively on D-lactose. For Ruminococcus, as expected, it grew in all 3 different sugars. And then the co-culture also grew. And if our hypothesis is correct, we would see that the Bifidobacterium would dominate this community in the presence of Ruminococcus. And from the sequencing data, this is indeed what we see. And this is actually kind of similar to the D-lactose situation, which led us to have this view where the Ruminococcus or another species encode extracellular fucosidase is breaking 2FL into D-lactose and L-fucose. Then the Bifidobacterium pick up lactose and can grow efficiently on this sugar, whereas fucose can be utilized by Ruminococcus or other community members for growth. So to further test this theory, we came back to this PT1. So as I mentioned to you earlier, this community did not respond to 2FL treatment because it is lacking extracellular fucosidase encoding species. So what if we add this species into this inoculum? Could we see something like this? And this is what we tested. So we took the PT1 inoculum and then we grew it at the 2FL. And as you can see, without the Ruminococcus added, you see the community structure barely changed and the pH is close to neutral. However, when adding the Ruminococcus, we can start really shift the community into a Bifidobacterium dominated community. And to just put the PT1 and FT1 side by side, we could conclude that when supplementing the microbiome matching 2FL and also Ruminococcus, you can start to reshape the preterm infant microbiome into a full-term-like Bifido-dominated community. So in conclusion, I demonstrated to you a novel mechanism explaining why different infant microbiomes have distinct responses to 2FL. And this study also emphasized the importance of providing microbiome-specific interventions. So I skipped a lot of details. If you're interested in our work, please check out our preprint. And I just want to close by thank my lab, my supervisor, Dr. Jill Benfield, and also our collaborators, Jennifer Donna's team, the Rubin Lab, and also the Morowitz Lab. With that, I thank you for listening, and I'll see you at the Q&A.